Welcome to this week's SciMon. I'm Jacques.
And I'm Paul of the Scientific Affairs team here at Illumina. Today we're talking about next generation conservation.
Specifically, we're talking about how next generation sequencing is helping in the restoration and conservation of archaeological, historical, and cultural sites and artifacts.
Cultural sites and artifacts are important because they make history real. They challenge and enhance our understanding of the past, and they can frame how we think about and how we act in the present.
But preserving or restoring these treasures is not an easy task. They're typically found outdoors where they are exposed to a series of physical, chemical, and biological factors. Even artifacts found in museum collections can require extensive conservation and restoration efforts.
For stone or wood artifacts, their surface provides an ideal habitat for the growth of microorganisms. Bacteria, fungi, algae, and lichens are all typically found on stone and wood surfaces. These microorganisms, over time, can break down the stone or wood by secreting enzymes and chemicals.
Sometimes these microbes can also help protect stone or wood, so it is important to characterize the microbial diversity on the surface of the artifact to help plan conservation or conservation strategies.
A nice example of this is a recent paper by Kimienti and colleagues. They used next-generation sequencing to profile the microbial diversity present on the surface of carbonate stones from the medieval church of San Leonardo di Ciponto in Manfredonia, Italy. To accomplish this, they first extracted metagenomic DNA from samples of the church exterior walls.
After performing 16S ribosomal RNA amplicon sequencing of the metagenomic DNA, they found that the community of microbes living on the church walls were composed of only 7 bacterial phylotypes. These were enriched in bacterial extremophiles, and those are microbes that are highly adapted to desiccation and UV radiation. They also identified photosynthetic algae.
In addition to harmful microorganisms, they also found species of Arthrobacter that could help to biomineralize the stone and protect it. The overall microbial diversity they identified could help in designing conservation and restoration strategies for San Leonardo di Ciponto.
In another example of this approach, Gutanowska and colleagues profiled the phylogenetic diversity of microbial communities found colonizing brick and wood artifacts in the former Auschwitz-Birkenau concentration camp in Poland.
The structures and artifacts in these camps are of exceptional historic importance. They serve as memorials of the tragic events of World War II and as symbols for the Holocaust. Because of the unprecedented historical significance of this site, intense conservation and preservation work is currently in progress.
Gutanowska and colleagues isolated metagenomic DNA from 8 wood and brick samples taken from sites located in the camp. They analyzed the samples with electron microscopy, metabolomics, and next-generation sequencing of bacterial 16S ribosomal RNA and fungal ITS1 amplicons.
Interestingly, they found higher microbial diversity in brick than in wood. Brick samples were colonized primarily by algae and cyanobacteria, while wood samples were colonized primarily by fungi. Most of the identified microorganisms are implicated in wood and brick deterioration. Metabolomics analyses of these samples confirmed that the activation of secondary metabolite pathways was associated with organic acid production and organic compound deterioration. Overall, next-generation sequencing helped in identifying 99 bacterial genera and 113 fungal genera from Auschwitz artifacts.
Previous reports used conventional Sanger sequencing methods to identify 8 bacterial genera and 23 fungal genera on wood and brick samples from Auschwitz. Next-generation sequencing captured more microbial diversity than Sanger sequencing and may provide more information to design conservation and restoration strategies. But that is enough for today.
We would love to hear your thoughts about our show and topics you would like us to discuss. To subscribe to our SciMon channel, watch more episodes, or get more information, click on the boxes on your screen.
Until next time, bye.
Bye.